I just want to first start off by acknowledging the grant that has made this all possible. I think Ruth alluded to that as well. It was a grant that was awarded to Drew Carmichael, Sinead Behind, and myself. That and the objective of the grant, which is sunsetting this year, but culminated in making this toolkit more accessible to all of you. And we took, did surveys of 13 sites around Mobile Bay over the course of the year, monthly sites, and with the objective of trying to understand the wastewater inputs and wastewater and sewage inputs into Mobile Bay, and from there identify the hot spots. And what we did here was a combination of classical microbial indicators, the Anaphylaxis, the E. coli, things of that nature, male-specific cultivation, combining with things that are more chemical in nature, stable isotopes and nutrients. I then combined that too with quantitative PCR, which was under the direction of Penny Dimitri here, that focusing on bacteriales, and then thirdly on eDNA, which is much more advanced, but also we hope that it's very tractable. And from there, combining these data sets, which is what we're in the process of doing at the moment. And so I'm going to turn this over to Penny to kick us off for the rest of the day, and I'll be here to, of course, answer questions and to assist in any way.
Thank you for the background. I'm going to continue with some background on Toolkit, and I just want to emphasize that throughout this whole presentation, throughout the whole day, if you have questions, please just stop me, interrupt me, raise your hand. I want you to get as much as you can out of this. So if you want to turn this into more of a chat, go for it. So we're going to start off with what is microbial charge stacking. I'm also going to refer to this as MST during the presentation and for the remainder of today. MST methods are used to detect fecal contamination. There are a bunch of different methods and they all vary in capability. So molecular MST methods are capable of distinguishing biological source. This means that they're able to tease apart human and non-human sources of fecal contamination. This is very important because in order to manage and mitigate fecal contamination, you need to know the source. You need to know who it's coming from. So where does environmental DNA come into play? What is environmental DNA or eDNA? eDNA is DNA that's present within the environment. So within water or sediment, it can be blood, tissue matter, fecal, bacteria, anything that an organism is shedding and is then present in the environment is considered eDNA. There are two ways to analyze eDNA. The first is metagenomics, which looks at all the DNA in a sample. If we break down the word metagenomics into meta, which refers to all, and genomics, which refers to DNA, we understand that it looks at everything within the sample. Metagenomics does not pre-select for any organism. So you're taking a sample of water or a sample of sediment, and you're looking at everything that is living in that sample, everything that's passed by that sample, everything that has shed DNA into that sample. The other way to look at an eDNA sample is called targeted sequencing or metabarcoding. And if we break down the word metabarcoding, we get meta referring to all again, and barcoding referring to target. So this can be used to target a specific community or an organism. For example, we can use the 16S rRNA gene to look at bacteria, or 18S rRNA gene to look at eukaryotes. So this figure right here depicts those two different ways to analyze eDNA. So starting from an environmental sample and extracting the DNA from it. In the top of the figure, you would target your sequences in the sample, and that's metabarcoding. So you're only looking for a particular organism or particular community, so you're only seeing that particular community. Or the bottom of the figure is metagenomics, where you're not selecting for anything. So you are looking at the entire community within that sample. You are looking at all of the DNA that exists in that environment. Thanks. So why would we want to use eDNA as a source tracking method? Other molecular MST methods, such as qPCR, have shown problems with watershed specificity and timer cross-reactivity. So this means that in order to use them within a watershed, you need to go through some tests to validate your timer sets to make sure that they actually work on your organisms within your watershed. Because eDNA metagenomics targets all of the DNA in your sample, I'm gonna hammer this point home. So if you get tired of me saying it, good, that means it sticks. Because it's looking at all of the DNA in the sample, you don't need any timers. So you don't need to identify potential fecal contributors in your watershed. And you don't need to give this a test to make sure that your timers work within your watershed. However, eDNA hasn't been fully implemented as a microbial source tracking method. So we created a toolkit to help. Yes.
I have one question. Like, what's the difference between qPCR and normal PCRs? Can you tell us?
qPCR is quantitative PCR. So when you're running the reaction, you add a fluorescent dye into your sample, and you can calculate the amount of starting DNA in your sample from that dye.
So basically, just I'm putting a dye, that means it's qPCR, and not putting a dye that's normal PCR.
It is run on a different machine. So the qPCR machine will take pictures of the dye after every cycle of the reaction. So that's how you calculate the curve, the amplification curve. And then regular endpoint PCR is run on a different machine, and you have to run it on a gel after, and it just shows you presence absence. Can't really quantify.
Thank you.
Any other questions before I move on from, that was the background portion. So we're gonna move into more of the toolkit now, if anyone has questions.
I'm gonna add one thing to that. The standard, it needs a standard curve, just like you would run any analysis with a set of standards. And quantitative PCR is great because we can quantitate, but you also have limitations to the standards, right? So anything below about 10 to the 4th targets or copies of that gene, you can't really detect, right? It's harder to pick up in that range. You're safely 10 to the 4th to 10 to the 7th is a great range for one to be in for quantitative PCR. The metagenomics takes away, of course, the quantitative aspect that allows you to detect things in a rare biosphere, a small, a small range. Yeah.
I was just gonna ask, volume, you say 10 to the 4 for what volume?
The targets per microliter.
Microliter?
Yeah.
Cool. Okay, anything else before we move on? Okay. I'm not gonna advance the slide, I didn't talk about the slide yet. Okay, so the eDNA search tracking toolkit has a website. So that little QR code at the top of your takeaway sheets, that will bring you to the website. And there will be workshop materials. So this PowerPoint that I'm going through is going to be posted on the website after the workshop. And there are also toolkit protocols. So there are full written detailed protocols for every step of the toolkit, and they are available on this website. So everything that we do during the hands-on demo part is shortened versions of that, but the full detailed protocol is on the website under the toolkit section. I put a little yellow box around it. That's where it lives. You can go find it later. It'll be there forever. Okay, now we're gonna get into the workflow of the toolkit. So I've broken up the workflow into 3 different sections. I also color coded it just because you'll have access to the PowerPoint later to make it easier. The 3 sections are sample processing, sequencing, and then bioinformatics and data visualization. So if we have any questions on the structure of the toolkit, let me know now. Otherwise, I'm going to jump into sample processing. Cool. Okay, so sample processing practices of the eDNA source tracking toolkit. The most important takeaway that I want you to get from sample processing for eDNA is sterility. Again, metagenomics picks up all of the DNA in your sample, so you really need to be sterile when you're processing these samples. If you don't, you can introduce your own DNA into the sample, and the sequencing will pick it up. You will find acid DNA in your sample. You're going to find bed DNA in your sample. And you don't want that, that's contamination, that's not what we're looking for, so sterile, sterile, sterile. We're not being crazy, we do see human DNA in there. The first step of the toolkit is actually collecting your sample. So you collect a water sample using sterile technique. So this would mean that your container should be sterile, you should be wearing gloves. And we suggest collecting a greater volume of water. So when we sample, we use carboys that held about 10 liters. They're in the back in the filtration stations, you can mess around with them later. We find that it's easier to collect a larger sample in the beginning than to not have enough sample to process later on. So that is what we found through our experience. After you collect your sample, it should be immediately placed on ice within a cooler and transported back to the lab. And filtering should occur within 24 hours of sample collection. Placing it on ice and filtering immediately is to prevent degradation of the DNA and to give you the best quality DNA from your sequencing that you can get. Back in the lab, you're gonna do a lot of filtering of water. So filtering equipment should be sterilized between samples. Again, sterility during every part of the process. And this is detailed in those protocols that are on the website. And this picture right here is what's set up on the filtering station in the back. So this is a peristaltic pump with inline filter tower setup. So what it does is it uses this tubing that runs directly from the sample container through a pump that's pumping the water through, and then through this crazy looking alien thing that's called a filter tower. But what it does is it has a filter in between those two, I don't know, alien tower things, and it's pushing the water through. So the water is going from the sampling container through the filter without ever touching the air. It's the least contaminated way that you can filter. It's also really nice because the pump pushes it through pretty nicely instead of waiting. Then once you're done filtering, you're gonna store your filters before extraction. And these sample storage suggestions go for all points of the sample processing, so filters as well as DNA extracts. Samples should be stored at -20 degrees Celsius to preserve the DNA, and freeze thaw should be avoided. This is when you're taking the samples in and out of the freezer. Both of these things are done, again, preserve the DNA to prevent degradation of that sample. Yes.
I believe that what I've seen on eDNA with eukaryotes, people have done the filtering in the field. And so is this just an example for maximum stability, or is this specifically for microbial?
Maximum sterility and also for greater volumes of water to get deeper cleansing.
Can you also filter your microbial samples in the field?
Yeah, like Starbucks filters, we thought about using them, but they don't filter as much as the large flat filters that were in the picture. So if you don't need as great of a volume, then yeah, filter in the field.
Yeah, I've seen people doing stuff in smaller volumes, trying to detect fishes from field samples, so.
Yeah, if you're targeting your eDNA sample, then you can go for smaller volumes. If you're doing metagenomics, it's best to collect a greater.
That's the recommendation. Okay.
Yeah. Thank you. Anything else? No? Okay. The next part is DNA extraction. So once you have your samples, you've filtered them, now you're ready to extract the DNA. Again, sterile space. This can be done within a fume hood, that's best practice. And you want proper PPE. And what I mean by proper PPE is looking like a lunch lady. So you want a lab coat, you want a face mask, and you want a hairnet. Because you're extracting your sample, anything that you release in that moment is in your extracted DNA. So again, we want to minimize contamination, minimize getting our own DNA and our own microbes into the sample. We also want to run an extraction blank with every set of extractions that we run. And this is a tube that you take through the whole process. You're adding all of the reagents, you're treating it exactly the same, you're just not adding a filter. So you're trying to see what is in the kit biome. So these are the microbes present in the extraction kit, consumables and reagents. So this extraction blank is going to pick up what's already there. and help you look at what contamination is present, because as much as we try to be sterile, something's gotta slip through it. So these blanks should be carried through extraction, and then they should also be sent for sequencing. And these sequences that are in the blank samples can be removed from the sample sequences that are bioinformatics. So you can control for contamination, you can sequence for contamination, and then you can actually do something about it. For DNA extraction, there are a few different methods that can be used. You can use a homebrew method, for example, phenol chloroform, or you can use a kit, which comes from a molecular biology company, and it has pre-made reagents, pre-made protocols, and you kind of just follow the steps and get your DNA sample. The DNA extraction station in the back is with kit, so the hydrogen powered water kit. So kits provide consistent recovery, whereas homebrew methods can help you get stuff out of other samples or lower biomass.
Am I late?
Yes, if you wouldn't mind grabbing a folder and a name tag and then coming and taking a seat, thank you. Some sequencing facilities can also do DNA extraction for an additional charge. The downside to this is that they're gonna sequence, or they're gonna extract it with a basic protocol. So if you have a tougher sample, or you need some optimization to your DNA extraction, they're not gonna do that for you.
Kenny, have you all tried different DNA extraction kits to see optimization or what kits are best for different examples?
Yes, before I started my sampling, I tried 2 or 3 different kits and a homebrew method, and I found that the hydrogen powered water kit provided consistent recovery.
And let me just say that my lab has done hundreds of sites globally over deep sea, extreme environments to coastal, and no 2 sites are ever alike. And I tell that to my students that come in. There's not going to be one magic kit that will solve all your problems, and that's why I always I never defer to a sequencing facility to go ahead and do the extractions for me. I'd rather optimize myself. So there are a range of methods that you can use. It depends on the amount of salts that are in your sample, the amount of humics, the amount of carbon, the biomass that you're targeting, the pH of your system. So having a good understanding, the characterization of your system is very important to start with, and talk to folks, scour the literature, give me a call. We're always glad to talk about these kind of things, but it's not ever one size fits all. The reason why we aimed for a kit in this particular case was that there was the right amount of biomass, which was very high, and there was a very high throughput, and of course, cost is a factor. The kits do cost more than a phenol chloroform extraction. But we had budgeted for that, to establish that consistency. And so we're not promoting this particular kit. There is it's definitely opportunities to get trials, from your vendors and things like that, and also seek out methods that work for your site. Humics and salts are 2 of your biggest enemies to DNA requirement.
Okay, anything else about DNA extraction? Okay. So after you extract your DNA, you should really check your DNA for quantity and amplification before you send it for sequencing. So the quantity should be checked with cubic fluorometer. We have one in the room, and it's part of the sequencing protocol. So if you get to that station, feel free to check it out. Most sequencing facilities actually require you to quantify your samples before you send them. So this is kind of like you have to do it. The amplification of the DNA, you don't have to do, but we really suggest that you do it. This is to check the quality of the DNA. So what you do is you run a basic endpoint PCR. You could choose some 16S primers and 18S primers, something general to make sure that you actually have something in your sample. And then you run it on a gel to make sure that it's amplifiable. At this point, if your DNA is not amplifiable, if it's not of good enough quality, you can run some cleanup steps. You can try diluting your sample to dilute any inhibitors, or if need be, you can re-extract. But this is a good stop point to check, make sure your sample is of high enough quantity and quality before sending it out for sequencing. Because sequencing is sequencing is the expensive part. So good idea to really check your samples and make sure that they're okay before you send them off. Okay, that's the end of sample processing. Questions before we move on to the sequencing part?
Do you find it hard sometimes, I guess, when there's turbidity involved with it? Because if you're using a set amount of water, do you guys use more filters to help that? Like, how do you go through that process?
So turbidity definitely decreases the amount of water that we can get through a filter. We try to filter multiple filters from a site, if we can't get enough water through it. Just to make sure that we have enough filter to go back to in case we have to re-extract. So we just try to collect, again, like that 10 liters of water is a really good amount, and we try to filter that whole thing.
How would that process change if you were doing the sediments?
If you were doing a sediment sample, again, sterile collection, freeze it immediately. You could also use liquid nitrogen or like a dry ice to freeze your sediment immediately, because it's, you know, worse than water. And then you wouldn't filter it, you just take it directly to extraction after you freeze it. And instead of there are specific kits for water extraction and for sediment extraction, so you would find a kit that has that is for sediment, or a method that is for sediment. And then you would extract it and again do the sequence or the quality and the quantity checks. So just different kit and no filtering.
So you said at the beginning to offer for Enterococcus and E. coli?
Say that again?
Enterococcus and E. coli is one of the targets, or just, that was an example? Because I'm curious, you're using a 90, so 90 centimeter.
Oh,
Water.
the 147.
Oh, 147, okay, that's right. And then they're using what, the apparel, water, the larger 50 mil?
Yeah, there are 5 mils.
5%.
So-
Okay, just curious.
Mhm.
Thanks.
Okay. Anything else?
You spoke on the replication.
Oh, yes, so you want to collect biological replicates as well as do some extraction replicates to make sure that what you're collecting is consistent and can be replicated and wasn't just a, human error or something that you were doing. So you can either collect multiple different containers of water at each site, or you can take if you have one of the larger containers that can serve as your biological replicate and just do multiple filters from that. And then when you're doing extractions, so you can't fit a whole big filter, or you shouldn't even fit a whole small filter into the extraction tube. One, to make sure that if something goes wrong during the extraction, you have enough left to go back to, and 2, so that you can do multiple extractions to have extraction replicates and make sure that those are consistent as well. So replicates for everything.
Do you have a cost or price sheet for, what it would take to get all the equipment to do everything up to this point? Or to do the workflow?
I have all of the,
Or with some of my work to
yeah,
do an envelope calculation for that.
I have all of the materials listed in those detailed protocols. And if it's from if I use a kit as an example, I give the link of that kit, but I don't have the costs listed. If that's something that people would like, I could make a sheet and put it on the Toolkit website. Okay.
When collecting the water samples, did you have any like specific like strategies or like where you collected them?
So if it's a site that I can easily reach the water, I would just make sure I was gloved up and put the carboy in the water. When you put it in the water, it's sterilized before you sample, but you still wanna like shake out the first few times you fill it to make sure that it's, wash it out with the sample water that you're gonna fill it with. If it's a site that you can't reach, like if you're kind of like over a bridge, there are horizontal water samplers that you can use, so they're like, I'm gonna describe to you what is in my head. It's like a tube and it's got openings on both ends and it's on a string. So you put it in the water and you drop a weight down and it snaps both sides shut. So then you can pull up a water sample and fill up your container. And again, you wanna flush it out with the sample water a few times before actually filling it. But that's how I would do something that's easily accessible and something that's a little further.
What about like collecting around weather? It's rain.
Yep.
Water, I mean.
Yeah, so, so for this project, we sampled every 4 weeks for a year. So yeah, some days it was super windy, some days it was super rainy, and we still went out, we still collected. It's a good idea to collect metadata whenever you're collecting your environmental samples. So we check the USGS gauge stations for like freshwater discharge, rainfall, and then we also brought along a YSI, which is an environmental instrument that can collect like dissolved oxygen, temperature, salinity of the water, so that we have all that information to kind of help us figure out the context clues to the sample that we get. But yeah, rain did not stop us. Anything else? Yeah.
So in the case that you had multiple samples, from multiple sites where you needed the horizontal collection tubes, to sterilize them between samples, did you guys put them in like all their equipment in like dilute bleach solution, or what does the sterility look like between sampling location?
Brandy has no concept about.
Yeah, it's like, I know that that's something that some folks use, but there's a lot of controversy around that, so that's why I'm asking.
Unfortunately, bleach can over sterilize, right? Because it can leave behind a residue that actually becomes a DNase or an RNase, and that's an enzyme that will degrade or deplete the DNA or RNA. So it's great for hospital that wants to be completely sterile in the, maybe their surgery field, but it's not good. Because it will leave behind, even with rinsing a residue. So simple ethanol works great to rinse, your instrumentation. Of course, we always autoclave anything that we can bring out, but you're right, if we're one of like the Van Dorn sample, we're not able to go back and re-sterilize it at home, but ethanol works great. And then, continuing to rinse very well with the sample itself, it's just fine. The key is that we don't want to leave behind anything, and that same thing with the RNase way that you'll see that you can spray on lab surfaces, and that will also leave a residue behind. So if somebody does decide to use bleach in their own lab, rinse it with water, rinse it with ethanol, try to get rid of that bleach as much as possible. It's very sticky though, and it does tend to get in places that you didn't intend to in the first place.
Maybe it's bleach.
In certain situations, if I'm having a hospital procedure, I definitely want them to bleach that place down for me. But no, in this case, it's too much. It's not a good thing. Anybody else have questions?
Okay, cool. We can move on to the sequencing part of the toolkit. So metagenomic sequencing of samples can be done by an external facility. Most of these sequencing facilities will list their services and their prices online. So here is a list of just a few sequencing facilities. This PowerPoint has the links to the websites. And then this is an example of the cost per sample. Sequencing technology can be long read, for example, Nanopore, Minaron, which is what we're gonna do today, or it can be short read, for example, Illumina. Long read sequencing technology means that the technology doesn't have a limit to the length of the DNA strand that it can accurately base call. Whereas short read has a limit to the length where the longer the DNA strand, the base calls decrease in quality and accuracy. So for short read, can also be called shotgun sequencing sometimes because you actually have to cut up the DNA before you go and sequence it to make sure that the strands are short enough for accurate reads. Before sequencing, all samples must go through library prep. So the protocols that we're going to be running through today are mostly library prep, and then you put the sample in the sequencer. It also includes some cleanup after library prep, quantification, and pooling before you put the samples in the actual sequencer. Library prep means that you're adding unique barcodes to your samples. These barcodes help to distinguish between different samples, so you can put multiple different samples within the same sequencing run, and then pick apart which sample is which based on the barcodes. The barcodes are also picked up by the sequencing technology. So for Illumina sequencing, it needs those barcodes or the adapters to attach to the sequencing technology to begin base calling. The samples are called libraries once adapters are added. So if you see the word library, it just means it's your sample with little guys on the end. That's it. That's all it means. And the short read library prep also includes the set that contains the DNA before you add those adapters. PCR. So we're gonna again do a little bit of that with the sequencing stations later. And by later, I mean now. So we have 4 hands-on demonstration stations that we're going to split up into.
20, make sure that you're using a freezer that is a lab grade freezer, not just a Home Depot freezer that doesn't go through freeze thaw cycles or frost free freezer. -80 of course doesn't do that, so it keeps it at cryogenic. So it's definitely more ideal if you have that accessibility. But just to say that not all hope is ever lost, I have gotten samples that have from RNA that have been in a freezer, in a chest freezer for 20, 25 years. I will never throw the baby out with bathwater. There's nothing that I think everything there's ideal, and then there's we're going to get it done. And we're going to see what we see. And so I'm just going to give you the ideal and the best practices. And then of course, never just throw something out because you think it just hasn't been stored perfectly. Just know your caveats, know how to later on. And that's the important thing with all of this is if you understand what each of these steps are doing and the science behind it, you can know how to count your data, know how to interpret your data, and how to both the successes and fallbacks of what we see. Thank you.
Okay, I'm going to move on. If there are no more questions to the last part of the toolkit, where we're going to talk about some bioinformatics and data visualization. So when you send your samples away for sequencing, what does it look like when you get data files back? If paired-end sequencing technology was used, so for example, over the next each sample is going to have an R1 and an R2 file. So for example, WTP12 has an R1 and an R2. So these are the forward read and reverse read, and they go together. You need both of them for paired ends. And they also end in .pasq, meaning that the file has sequence and quality information. So these are the first few lines of one of the FAQ files. Looks like a bunch of junk, but it is actually the sequence information and the quality information. information, so there's a lot of meaning to these files. This bioinformatic processing of sequences can be done locally, meaning through like your own computer can handle it, or through a high performance computer, for example, the Alabama supercomputer. The Alabama supercomputer can be accessed through your local computer through something known as a terminal, and I'm going to pull up one in a few slides to show you guys what it looks like. Some sequencing facilities can also run basic bioinformatics for an additional charge, but again, just like with the DNA extraction, it's a general, basic pipeline that they'll run it through. They're not going to optimize it for your needs. So the first step of bioinformatics when you have your raw sequences is quality control, which involves quality analysis. This can be done with software such as FastQC. This is an output of one of the FastQC graphs. So when you run FastQC on your samples, it will output a bunch of quality graphs based on different parts of that FastQ information. So this is a quality score graph, and what we can see is that the majority of all of our sequences are good quality. We have one that is not good quality. So it's picking apart our sequences and based on that quality information, giving us a summary of what that means. Once you look at quality analysis, you trim your sequence ends. So you can use a software such as Trimming for this. And what you're doing is you're trimming off those barcodes that were added during library prep, because that's not part of your sequence, it was stuff that was added to your sequence. You can also trim the bad sequences from your sample. So for this, I would want to get rid of that red sample because it's not good quality. And then I would be left with just the green ones that are good quality. Okay, after you look at the quality, you're going to assemble your raw reads into contigs. And I'm going to walk us through this figure here. You can use software like Spades and MegaKit to assemble your raw reads, which are your shorter sequences, into contigs, which are longer strands of DNA. So you take your sequences from your raw reads, you compare them, and the software outputs it into a longer DNA strand. Once you have your contigs, you can annotate with taxonomy. For example, you can use a 16S rRNA gene or 18S rRNA gene, and functions, you can use mRNA. So when you take your contig, you annotate it with known databases, then you can identify different genes that are in your sample as well as species. So I just threw a workflow at you that has a lot of different steps and a lot of different software. So thankfully, there is something called Envio. It's a free platform and it integrates bioinformatic workflows. So instead of running all of those different software individually, you can run an Envio workflow, and they have different ones for different things. So they have a metagenomics workflow, and what this workflow will do is it will take your raw sequences and output the assembled and annotated contigs. So it'll take your samples from the first step, run all the other steps behind the scenes, and output the last step for you. It's beautiful. It also has some really nice data visualizations. So I'm going to show you how this works. These are two examples of data visualizations from Envio. And what we can do, this is a terminal. So this is what I was talking before. I'm logged into the Alabama supercomputer on this local computer. And what I've done is I've loaded the Envio module, and then I've moved into the folder where I have my sample files. So I've run these samples through the Envio metagenomics workflow, and I'm going to show you the line of code to generate these data visualizations. And thank you to Cassie. These are mobile Baykeeper samples that we collected at the Mountain Dog River. So real data from real samples that have gone through everything that you did today.
No, thank you, Penny.
There we go. Okay, so I'm going to copy and paste this into here. And it's going to take a little second. It's slow, but it's going to run this and it's going to give us an output that I can paste into a web browser to generate that visualization. And this is called an AMB profile. So what it's doing is it's taking the config data of your assembled configs and all of the annotations. So any databases that you've annotated with, it's taking all of that information and outputting it in a circular genome chain.
If you were doing this on your standard laptop, not the supercomputer, how long would it take to go through this roughly?
It was so short, it depends on the number of samples that you're working in. If you're just working with those samples, it's just that quick.
Okay,
If you're working with a large number of, a few hundred, samples, in this case, that's why, she's working on this.
Yeah, so it's not time, it's more memory storage. Okay, so what I did is I pasted that into the web browser and I clicked draw in the bottom corner. And this is our sample. So it's interactive, but it's slow because this is a really big sample. So you can zoom in to try and see different things. I zoomed in, it'll take a second. And then you can highlight different parts of it. In the middle of this graph are all of the contigs within the sample and how they assemble together. And then the outside shows the different databases that were used to annotate the sample. There are also different tabs up here. So you can search, for example, you can search through the functions of the databases that were used to annotate. So if I want to see how many times nitrogen popped up, 4,000, almost 4,000 results of nitrogen within my functions for the sample. So that is one example of an NGO data visualization. And then if I go back here and go back to the command line, I'm going to show you the other visualization. So what I'm doing is switching directory into that, and then running this code to generate what's called config stats. So this is an output of the statistics on your config. So generally, what organisms are in your sample, what genes did they find? And that's what this looks like. So again, it's interactive, so you can zoom in and see, let's see if I can make this clearer. This output on top is all Archaeobacteria and Protista, and it has the length of the contigs and how many contigs for each of those size groups. And then on the bottom, it has the genes for each of your taxonomy groups. And it also has a printout of this for further information on the config stats, like the length, the annotation information, approximate estimations of genomes. Yeah.
Can that command line be found on like the NVIDIA website?
No.
Like a.
Or do you mean the script that I used to run or the terminal?
The script.
The script, yes. It is also in the detailed protocols that I'm putting on the website. So I have one for specifically running NVIDIA workflows and running them in the supercomputer. So exactly the script that you need to run. Yeah.
And so will this also work with eukaryotes as well, or is it basically, are you just pulling from databases that have like prokaryote?
Right now, I pulled from prokaryote databases, so that's why it's mostly showing or only showing Archaeobacteria and Protista, and then at the bottom, it doesn't have any eukaryotes. But I'm going to run it against other databases, and you can choose which databases to run it against. You could do it against a general database, or you could even do some like more specific curated databases. So whatever you want to answer your questions.
So, just a double check, it'll take all your FASTA files, for your versions, match them up for you, clean off the ends, look for mess ups, like that, then run on database, do all that for you, and then run it out.
Yeah.
Okay, very nice.
Yeah, you can program it to choose which databases you want. The scripts that I have in my protocols are for certain databases, and I tell you which databases I've asked it to do, but you can ask them for different databases, but it takes it all the way from cleaning and pairing and annotating.
Right, and all the back, so well, it'd be nice not to go through that, route, where you have to take all that data, and then it'll put your King file and all that, in there. Does it go that far?
It does have some outputs. You might need to run, I'm working right now on a Kraken 2 script with Kraken to try and see if I can get those outputs in the table format.
That's awesome, because less steps, nice, okay, thanks.
Yeah. 80.
I use Anvil for a lot of my PhD research, and there are a lot of very helpful tutorials on the Anvil website. And if you still have questions, the developers are extremely friendly and are happy to answer any questions if you reach out to them directly.
Thanks, Katie. Okay, any questions on those Anvil visualizations? One more slide, and then we're gonna move to wrapping up. Okay. Switch back. On my other thing. Somewhere. Okay. Last thing is some statistics on your samples. So if you collected environmental data or you have some metadata alongside your eDNA samples, you can compare your metadata to your taxa. An example of this would be using the Vegan Community Ecology package in R. And this picture right here shows some different environmental parameters compared to different markers or taxa. And this can help you tease out which relationships are significant and what's driving that community structure. In R, you can also use the decontam package. So I harped on blanks earlier. This is where it comes back into play. So after you run your samples to the bioinformatics workflow, you can run it through this decontam R package, and it'll compare your blank sequences to your actual sample sequences and help you identify what is contaminant and what is real. So final quality control. That is the last part of the toolkit. If anyone has questions on that, feel free to ask. Otherwise, we're going to move to the wrap up. Okay, moving to the wrap up. We have a 10-minute wrap up question, which is how can eDNA source tracking be applied to your own work? So I would appreciate it if you guys could take the next 10 minutes, you could talk to your neighbors, you could talk to us, but if you could think of some ideas, some thoughts, and if you could jot it down at the bottom of your takeaway sheets, that would be wonderful. The Toolkit website is going to house all of the materials, so the detailed protocols are already up there. I'm going to add the PowerPoint and the recording of the audio up there, so you can reference it later. There's some other materials on the other tabs if you need to check it out.